Okay, good morning, everyone. Welcome to Masonic Cancer Center seminars. Today is April 15th, 2025, tax day. I hope you have your taxes in. My name is Emmanuel Antonarakis. I'm the Associate Director for Translation at the Masonic Cancer Center. Today, we're going to do something different. It's going to be a bit of an experiment. As many of you know, some experiments are not successful, they are not repeated. And some are successful, and then we try to take credit for them and then repeat them in the future. And I just want to give an introduction about today's session and why we decided to do it this way. About 2 or 3 months ago, Carol Lang, the Associate Director of Basic Research, reached out to me and said, how do we make translational research fun again? And it was a really interesting question. I thought we were always making it fun, but it turns out that there have been so many pressures on scientists and clinicians, even before the new administration, that we kind of forgot that this should be and can be a lot of fun. And so it was Carol's idea. She said, let's do Masonic Cancer Center seminar, which is less didactic and more like a town square, like a storytelling. And let's invite 3 people who are really good at translational research, have succeeded, and they can sort of tell us, this is how I did it type of thing. And I said, Carol, that's a great idea. Would you like to be one of the 3 speakers? And she said, no, I don't do translational research well. And she said, do you want to do it? I said, no, I don't think I do translational research well. And she said, well, who else then? And we brainstormed and we came up with these 3 individuals that you will be hearing from today. And each one of them, I think, has been very successful at translational research. Now, it's interesting, you can define success in different ways. Sometimes there are successes that come from failures. You learn things along the way. And at the very least, new interactions and collaborations are forged, which are more important than even the goals of that one project, because they can be applied to other things. So when you hear from these 3 people today, it may not always be about a nature or science paper. It might be about the journey and what was learned along the way. But I think each one of them will hopefully inspire you, and we want this to be very interactive. So what I've asked each of the 3 speakers to do is to present no more than 3 to 4 slides. I think they've all adhered to that. Maybe one of them cheated. And then we want each person's remarks to be informal and to last about 10 minutes. And I think what we're going to do is we should hear from all 3 of them in a row. And then at the end, we're going to bring 3 chairs up to the front, and we're going to have an open discussion like a town hall. And we want this to be extremely interactive. It'll be quite boring if you all sit there without asking anything. So as these speakers are telling you their stories and their journeys with one translational project each, think about something that you might want to ask them, and you're allowed to ask them anything, even personal things or how that interaction started. It doesn't all have to be about the hard science. And we just want to help everyone in this audience realize that there are many ways to do translational research. There isn't one way. And a lot of the didactic courses that we have, they're great in general, but they don't give you a specific example. So I'm going to introduce all 3 speakers now, and then they'll be coming up one by one, but I'll do the introductions all at the beginning. So the first speaker is Dr. Melissa Geller, professor in the Department of Obstetrics and Gynecology and Women's Health, previously Division Director of Gynecologic Oncology, and currently Associate Director of Clinical Research. She'll be talking to us about an interesting immunotherapy approach for ovarian and other solid tumors. After that, we'll be hearing from Dr. David Largaespada, professor in the Division of Pediatric Hem-Onc, co-director of the Brain Tumor Program in the Brain TWIG, as well as currently interim deputy director of the Masonic Cancer Center. And finally, Dr. Doug Yee, professor of medicine and pharmacology. He has a dual appointment. He's also currently the director of the MNCCTN and breast cancer medical oncologist, and up until recently, the prior director of Masonic Cancer Center. So with that introduction, I'd like to invite Dr. Geller to present her 5 slides, and welcome, Melissa.
Thank you very much. Good morning, everyone. Thank you for, I'm going to start my little timer here. Thank you for coming this morning and navigating the traffic that seems to be a major issue. Success, as Dr. Antonaraka said, it comes in many forms. And I think, as a clinician, so again, in gynecological oncology, I never meant to be a translational researcher. I think it sort of started when, and I look back at Lauren Benzik, because she's been with me through this entire process, that I was working in a lab, we got an American Cancer Society grant, we got money. My mentor, who I was working with, left the institution, and all of a sudden I was like, what am I going to do? I have this money. I don't exactly have a mentor in my lab anymore. And so it just happened that way. It fell into my lap. And luckily I had Laura and many other researchers stay with me. And so we've been successful. I say from day to day, you never know what's going to happen, especially now. And I think that's how I started this career. And you'll see here this cartoon, it says it's nothing a few stem cells and another 75 years of research can't fix. And I'll tell you, it's you never know what's going to happen next. And there are days that I leave the lab or I leave lab meeting feeling like this. But overall, it has been exciting. So I'm going to start with the story about it. Really, when you look back at it, was probably 2012 when we first were contacted by a company called Altor. And this is why it says Alt 803. So this is a molecule that they came to us with. Jeff Miller really instituted this relationship with them. And IL-15 is a naturally occurring cytokine. It's responsible for maturation of NK cells, proliferation and priming of them. And we really started getting interested in this because of Jeff's work. And I'll show you his first paper that came out was a first in human study, phase 1 clinical study of using IL-15 superagonist, so this Alt-803, to treat relapse after transplantation. And so basically the gist of this is that patients tolerated it, it went well in a clinical trial, and that basically they were able to activate NK cells without stimulating regulatory T cells. And as most of you in this room know, regulatory T cells really are immunosuppressive. And so this was the first paper. So when we, Jeff said, well, what about in solid tumors? Should we try something like this? So we went to the lab, in our lab, and this was the first paper that we published looking at the use of this Alt-803, which originally was N803, to enhance natural killer cells in the ovarian cancer setting. So basically went to the lab, showed that we could produce and really help NK cells proliferate within our mouse model. So after that, we said, well, we started going back into the clinic with this. And this was a long-term relationship. And I have to say, it was a small company, easy to work with. It is not so easy to do this with larger companies. And we were really lucky because Hing Wang, who ended up leaving Nant West, really wanted to engage us and say, you know what, we want to make you the first clinical trial site. I was interested, Jeff was interested, and that's really how this relationship started to work. So I would say smaller companies, much easier to deal with than the bigger, larger pharmaceutical companies, because they are invested and they want this to work. So what we did is we started a clinical trial using this drug, this Alt-803, in patients with advanced stage ovarian cancer who received debulking therapy, underwent chemotherapy, and then went on to receive maintenance therapy with this drug. We gave it this IL-15, what we call the superagonist, both IV as well, or I'm sorry, subcutaneously as well as intraperitoneally. And we treated about 10 patients on it. So it wasn't it was a pilot study. But what we then did, and I didn't even really know this term before we wrote the paper, is we reverse translated. So we took all of these samples that we were collecting. So we collected peritoneal fluid, we collected blood, and we collected tumor samples from these patients. We went back to the lab and said, okay, why are these NK cells only persisting for 2 weeks? So we got about 2 weeks persistence with this administration. And at that point, we started investigating. And as you can see here, what we did is we saw upregulation of both PD-1 and PD-L1 on our NK cells and within our tumor. And so we started to combine the Alt-803 with immune checkpoint inhibitors. And this was really exciting for us because I don't know how many of you know in this room, but ovarian cancer, there's no indication for immune checkpoint inhibitors in ovarian cancer. We've had very poor results, less than 15% response rates. And so we were excited by this data. And as you can see, this is a killing assay here, basically looking at the NK cells alone, plus the N803 or the ALT803, the superagonist, followed by avelumab, which is a checkpoint inhibitor, blocks PD-L1. And when you combine all three, we really saw better results. And this again was published in our findings. So what we went on to do after that is, and this was an interesting little twist, NANT was like, okay, yeah, we want to do the study with you combining checkpoint inhibitors. Well, then they decided to pursue in bladder. They got the FDA indication in bladder, and they weren't so interested in ovarian cancer. Also because avelumab was done in prospective randomized trials in ovarian cancer, got poor results, not in combination with this ALT803. So we kept going to the drug companies. We went to Merck, we went to multiple, said, give us a drug because we can't get pembrolizumab in ovarian cancer. We can't get a checkpoint inhibitor. In the meantime, Hing Wang, who I worked really closely with at NANT, ended up spinning off his own company called HCW Biologics. And so this is the current drug that we are using. And so this is, again, an IL-15 kind of superagonist, but it's got a different moieties on it connected by a tissue factor, soluble tissue factor. These are TGF-beta receptors, and basically it's the TGF-beta sink. And so it kind of gathers up all that TGF-beta, which is a immunosuppressant. So we were excited about this. He was excited about this. And so we said, okay, let's do a new trial. So this is our current phase 1 clinical study in advanced stage ovarian cancer. So I'm sorry, not just ovarian, all solid tumor trials. So we brought this to our Developmental Therapeutics Initiative group. And this is a group that we have at the University of Minnesota dedicated to early phase clinical trials. And the nice thing is we were able to bring these patients in as an outpatient status. They're dosed once every 3 weeks subcutaneously. Then they were given multiple cycles. We collected tumor, we collected blood, we collected we were able to collect all of these throughout the first initial 2 cycles of these patients. The primary objective of this was a dose escalating, very typical for a phase 1 clinical trial. And the secondary objective was safety and feasibility. And so this was our initial trial. And what I want to show you, and I think this is important for everyone to know, is the study timeline. This takes a lot of time. I mean, this is like not something that these are things I do at night. These are things I do on weekends. This is not built into our usual everyday life of being a clinician and a surgeon. But the study timeline, and this is where I have to thank the clinical trials office. This was one that our goal was to open this in less than 90 days. And you can see all of the steps that are required to open such a trial. So ultimately we opened in about 73 days, which is good. And we want every single trial at the University of Minnesota to be open this quickly, and it doesn't happen. But this was one where we really pushed it and we were able to open. So if we have, if we can do this on a day-to-day basis, this is what our goal is. But you can see all the steps it takes, and it's the back and forth between, the Cancer Protocol Review Committee, the IRB, you have to have beacon orders written for this study. So it is a lot of work. And this is what the clinical trial office does here at the university to make this happen. You can see here, these are the demographics of the people that went on our trials between April 1st, 22 to January 9th, 24. We accrued 18 patients. And so the beauty of this is in this phase 1 early clinical trial setting, we have a list of at least 150 patients at one time who are looking for early phase clinical trials. And we don't have enough of these trials. I mean, we're constantly juggling spots. So they may, this was only open at our institution, but we have other phase 1 clinical trials that are open outside, and they give spots away. So we're constantly vying for a spot for these patients. But we did really well in accruing to a all comer, basically clinical trial setting in the Developmental Therapeutics Initiative. So just to give you a sense of what's next, and so, what we came out of this trial, and this is being sent, we're in the midst of trying to get this published right now. It's been sent in. we found the recommended phase 2 dose of this from this clinical trial. We found that NK cells and T cells really robustly expand and are activated. We can turn a cold tumor into a hot tumor with this drug. And we really, at least in the ovarian cancer population, saw some really stable disease over a longer period of time, which is encouraging. So we've gone back now and we're working with the company to design the next trial. This is going to be in recurrent platinum-resistant ovarian cancer patients. This is the other thing I tell you, these companies are constantly changing these drugs and they're constantly looking for something better. So as soon as you get this data published, there's a new molecule or they want to work on it. So it's really critical to develop, I think, really good relationships with these companies, because if you stick with them, they want to work with you. And if you work fast, they want your studies to be open at this institution. And we've struggled with that in the past because of the slow timelines, contracting, things like that. But it's so critical for making these investments. So basically, this is a new drug that they're coming up with, different protein scaffolding rather than the soluble tissue factor domain, and really stronger potency. So that's what we're hoping to use in combination with checkpoint inhibitors for the next trial. So I think overall, hope it gave you a little bit of a sense of what this looks like, and I'll answer any questions at the end. Thank you.
All right, David has an interest in neurofibromatosis type 1 and 2, and especially type 1 and the tumors that develop in these patients. And he'll talk to you about a new synthetic lethality for NF1, peripheral malignant nerve sheath tumors.
Yes, thanks. All right, so I thought today I would speak more from the perspective of a PhD research scientist getting into clinical translational research, and sort of what I think about that. So of the three speakers, I'm the one today who doesn't have an MD. So when I was a postdoctoral fellow in the last century, I got interested in NF1 for completely random reason, which is that I was working on slow transforming retrovirus that could cause acute myeloid leukemia in laboratory mice. And it turned out that in a subset of these leukemias, the provirus had integrated in and inactivated the NF1 gene. And actually this work helped clone the NF1 gene. And so it was a total accident. And it turns out that if a person inherits one mutant copy of this NF1 gene, then they're afflicted by this common cancer predisposition syndrome, neurofibromatosis type 1. Now, one of the types of cancer that these patients get with increased prevalence is are myeloid leukemias, both acute myeloid leukemia as well as a chronic form called juvenile myelomonocytic leukemia. And so I started working on that and got an interest in NF1. Over the years, I decided to start working on more common manifestations of disease in NF1 patients. They're prone to brain tumors and peripheral nerve sheath tumors, so glial tumors in the brain and in the peripheral nerve sheath. And that's what's diagrammed on the left-hand side of the slide, is the progression of these peripheral nerve sheath tumors. So the patient with NF1 is heterozygous for a loss of function mutation in NF1, loss of the wild type allele in a rare Schwann cell or Schwann cell precursor can lead to a benign tumor called plexiform neurofibroma, where the neoplastic cells are these NF1 deficient Schwann cells. That can progress to a pre-malignant stage with loss of CDKN2A and B, and then that can progress further to malignant peripheral nerve sheath tumor that we got interested in. So work on NF1 syndrome is probably the one constant in my career. I was poorly trained in the sense by Neil Copeland and Nancy Jenkins, in the sense that our lab has worked on a lot of different things. But my advice to you as a PhD research scientist would be at least have one thing that's a common theme that runs through your work. And for us, it was NF1 syndrome associated problems and tumors. The other thing I would say as a piece of advice is that get to know the patients and clinicians who are involved in this disease. So we've been super fortunate to work very closely with Chris Martell, who runs the NF clinic at the Fairview University Hospitals and Clinics. He really, I learned this too late in my career, that as a PhD scientist, you should try to get joined at the hip with a clinician who has an interest in the disease you're working on. This is super important for many reasons, but Chris introduced us to families affected by this disease. Many of them visit the lab, they raise money for the lab, they motivate the people in my group, and really put a face to what we're trying to accomplish. And by talking to them and by talking to Chris and other clinicians, it helped shape our research too, which has been really important. So I just told you about how malignant peripheral nerve sheath tumors develop. What they're notable for is what they lack. So they develop by the stepwise loss of tumor suppressor genes, NF1, CDKN2A and B, then either EED or SUS12, critical components of the polycomb repressor complex 2. So what's happening is that you're getting the loss of these barriers to transformation. What you don't see in MPNST is they're not notable for recurrent activated oncogenes that we can easily identify. So a rare subset of them will have activated BRAF mutations in the non-NF1 associated MPNST, but there isn't anything obvious that you could use as an inhibitor to inhibit an activated oncogene. So a long time ago, we got the idea, well, let's see if it's a loss of all these tumor suppressors that's happening. Let's see what drugs might kill the mutant cells and not the wild type cells. So it's a synthetic lethality idea. Perhaps the loss of these tumor suppressor genes uncovers sensitivities that these premalignant and malignant Schwann cells have that normal cells don't have. So that was the whole concept. And so Kyle Williams joined my lab, and he took immortalized human Schwann cells and made isogenic derivatives that we could use in CRISPR screens and drug screens that we've done to try to find sensitivities. So he used CRISPR to do that, and he made these NF1 knockout Schwann cells. He did a very large small molecule screen, over 11,000 drugs, including all FDA-approved drugs. So part of our idea was maybe we can repurpose drugs that are already out there for use in NF1, and then tested those drugs in mouse models for peripheral nerve sheath tumors. And he found almost 30 drugs that were better at killing NF1 deficient Schwann cells than wild type cells. And he took 5 or 6 of those and validated those in mice. To our surprise, all of them worked in mice, just like they'd worked in vitro. Felt super lucky about that. And then he started testing combinations, some of which I think are really exciting. And he's published that work recently. All right. He went further and is preparing a paper on where he's also knocked out SUS12 and modeled the malignant stage of the disease and found additional drugs that work when PRC2 is lost from these Schwann cells. All right. These drugs included histone deacetylase inhibitors, azacitidine, aurora kinase inhibitors. What was good about this is that some of these drugs are FDA approved for other types of cancer. And so that meant we could go forward quickly. And so what we found is that a combination of an HDAC inhibitor and a MEK inhibitor show synergy in vitro and in vivo versus MPNST. And we use a whole variety of model systems to show this. And it's a very robust finding. And this led to a new clinical trial that we just opened in January that combines 2 drugs, mirdametinib and vorinostat for PRC2 deficient MPNST. And Melissa mentioned working with a small company. One thing that helped was that mirdametinib is a company is a drug from a small company called SpringWorks, and they've been easy to work with, and they're providing free drug for this trial, which is really critical because it's a newly FDA approved drug. for the benign stage of the disease, but it's expensive. So that's really my experience, and I hope some of that resonated with you. This slide just shows some of the sources of support that we've had for all this research. All right.
Great.
All right, thanks, Doug.
So this, Melissa said her story is a long story. This is a lot longer story. And it is actually kind of, I don't know, I think it's a tragedy in 3 acts or 4 acts, but at any rate, and it's very different than the last 2 stories you heard. So my translational lesson number 1 is to become, and this is my case, known for something. So the thing we were known for is inhibiting IGF receptor signaling in cancer. Just Melissa started out with a story. How did you get to this point? When I started out as a postdoc / medical oncology fellow, I got interested in breast cancer because we knew targeting one protein, the estrogen receptor alpha, resulted in tremendous therapeutic outcomes. Everybody at that time said there must be something better. So that's what we worked on. This is a cartoon from a journal article and a review that Paulie and I put together in 2006. If I remember right, to Paulie, this was like the most cited article out of that journal that year. So that tells you the last time IGF receptor signaling was a hot topic. But just, I need to tell you about the system a little bit, just to go over some key points. Point number 1, IGF receptor is a transmembrane tyrosine kinase. It's a heterotetrameric structure. It has high degree of homology with the insulin receptor. And there are 2 forms of the insulin receptor, fetal and adult isoform. They can form heterodimers. Because of the heterotetrameric structure, they require interaction with a ligand to be activated. So unlike some single family tyrosine kinase transmembrane receptors like EGF receptor or HER2, they can't activate without ligand binding. Once they do that, they initiate a bunch of signaling pathways, and that leads to the bad cancer phenotypes. In addition, in normal physiology, you and I have hundreds of nanograms of IGF-1 and 2 circulating at the moment. They're all complex to binding proteins. There are 6 binding proteins. They have high affinity binding to IGF more than they do to the receptor. The principle is when you're traumatized, like burned or an accident, and even during pregnancy, you release ligand from the binding protein. So that's when we spent the first part trying to sort out all the important components of the system. We also, 10 years before this paper was published, we collaborated with a company called Synergen, and this is a translational lesson. And this translational lesson is primarily for the trainees in the room who are thinking about going into biomedical industry. It's both promise and a cautionary tale. So Synergen was a company that was making neutralizers of protein action. And the first thing they made was an IL-1 receptor antagonist, and they fused it to an immunoglobulin and said, look, we have this IL-1 neutralizing agent. Now, where do we test it? And they said, okay, let's test it in sepsis. That trial failed miserably. And in fact, people got the TNF-alpha receptor, IL-1 receptor antagonist had worse outcomes. They had higher mortality. They're not getting a drug. That's not going to do wonders for your stock price. And they crashed, and they were by Amgen. Amgen then turned that drug into a rheumatoid arthritis drug, and it was one of the first rheumatoid arthritis drugs that was actually worked. And it's, the disease-modifying drugs that now are popular in RA came out of that. So the cautionary tale for trainees is that the scientists who were working on this drug were fabulous. They cloned it, they made the protein, they did all the PK, but I want to be kind about this. They functionally lost their jobs because somebody at a higher level made a bad decision. So to me, that's a part of industry, is that, you have to be connected to a company that's making the right decisions, and they didn't. So anyway, the reason I told you that story, the other thing they were making was an IgA binding protein that was pegylated and had high affinity for the ligands. So in 1997, before I came here, we published that paper showing that, yeah, neutralization of IGF's ligands actually was effective. And the model we used actually was ovarian cancer. So if you neutralize IGF ligands, you can inhibit ovarian cancer cell growth. And we left it at that. So that's that story. And the story is that, yeah, maybe neutralization of ligand action would be a good way to do this. Now, the rest of the industry was focused on receptor antagonism, monoclonal antibodies, tyrosine kinase. Fast forward 20 years, all the monoclonal antibodies failed in cancer clinical trials, and none are being developed. The tiny silver lining of this story is that if you watch TV and you see TV ads for thyroid eye disease, and they talk about a new drug for thyroid eye disease, it's the bulging eye in the house, that's the commercial I represent. That's an IGF receptor antagonist. That is teprotumumab, an antibody that Roche Genentech developed. They dropped it, a startup actually bought it, and the startup made it into a drug. And now, we just came back from an IGF receptor conference. Now everybody wants to look at IGF receptor again in thyroid eye disease. So anyway, maybe it's coming back a little bit. But I want to tell you a story about neutralization. And the story about neutralization is zentuximab. It's an IGF ligand neutralizing antibody. So in 2006, around the time we published that, sometime after we published the neutralization paper, I was invited to Vienna, the home of Boehringer Ingelheim, the company that makes this drug, and the role of IGF signaling to talk about the role of IGF signaling pathway in cancer. So that's a translational lesson number 2. If you get to be known for something, people will seek you out, and you have a great opportunity to visit fabulous places. One of my colleagues calls this the world of oncotourism, but it is true. The biomedical international companies have headquarters elsewhere, and you have to go to them to talk about the thing. So they asked the question, and this is the best of my memory, should we develop this drug? I said, it's a great idea. It's completely different than what everybody else is doing. There was one other company that had a drug at the time that did the same thing. So they went ahead, and I thought they forgot about it. But 8 years later, I was invited to London, another fabulous oncotourism place, to go to an advisory board meeting and talk about zentuximab and design a clinical trial. So the clinical trial showed that zentuximab worked very well in mouse models. One of my colleagues, Michael Pollack, had showed that very clearly. So just like we had shown with IGF binding proteins, they showed neutralization of ligand was very effective. And then in 2017, more than 10 years after their initial contact, they asked me to serve as the steering committee chair and coordinating investigator for an international phase 1b trial in combination with CDK4/6 inhibitors. They were doing another trial at the same time. I showed you all those downstream signaling pathways on the last slide. One of the downstream signaling pathways is PI3 kinase. They were doing a combination trial with everolimus, a mTORC1 antagonist at the same time. And I got asked to do this other trial with CDK4/6 inhibitors, and it was done in breast cancer. So the bad news was that their everolimus trial failed. We all got a Dear Investigator letter roughly in 2021 that said there was no benefit for adding zentuximab to the existing therapy of everolimus and exemestane, and so we give up. And this is what companies do, I think. Once they get a whiff of things aren't working correctly, they stop. Another warning, so just like my Synergen story, there was a great team working on zentuximab. We were still conducting this trial. As soon as it stopped, it kind of ended. It kind of ended our communication with that team. They finished the trial. So this was the trial, and I realize this is a complicated slide, but it's a very complicated phase 1 trial. And I'm not going to go into gory details telling you all the different arms and why all the different arms were done. I will tell you one thing. If you're ever asked to do this, try to dissuade them from doing an international phase 1 trial. We had sites in France and Spain, the US, Japan, and Korea. And I'm just telling you that because finding a time for a conference call where everybody can meet every week to talk about a phase 1 trial was like terrible. So I think we met like 9 o'clock our time. So we had sometimes very early for Europe and not that late for Asia. But anyway, I wanted to show you the highlights of the trial. If you look at the cohort D
D2, I believe, D1, I believe. This is with abemaciclib, zentuximab, and fulvestrant, an anti-estrogen. This is what we call a waterfall plot. And you see with the cutoff here, there was reasonably good activity in that subgroup. Now, of course, this is a single arm phase 2-ish 1B trial. So you can't say this is adding anything to the standard of care. But certainly my colleagues at Boehringer Ingelheim at the time said, yeah, we should pursue this. But the company decided to pull the plug. So my hope is that some venture capital firm will swoop in and buy this drug, and they'll buy this drug to develop it in thyroid eye disease. Go ahead, develop it in thyroid eye disease. That'd be fantastic. At the same time, give us an outlet to continue this trial in breast cancer. So at any rate, that was a long story and a translational pathway. It's kind of, I think, a tragedy because I actually do think this drug has some activity. We need to do further studies to actually figure that out. But at the same time, you're always at the whims of business and business entities and what they decide to do. So that's my story. And I guess we're going to all take questions.
All right, we're going to bring 4 chairs to the front. How many microphones do we have?
We have 3 microphones without really one up
Yep.
front with the speakers.
Perfect.
And then Laura and I will work either end.
Thank you. While we're waiting for the rest of you to come up with your questions, I'll start with one. And I'm going to ask the same question to all 3, and then you can respond in your own way to the same question. One of the things that I started to realize when I was doing translational research and coming out of my own comfort zone is that there was always a new skill or a new knowledge that I had to develop for each collaboration. For example, when I first started working on the androgen receptor, I decided to go to a andrology conference that was run by endocrinologists. They knew everything about androgen excess disorders and androgen deficiency disorders outside of cancer. When I then started working on DNA repair deficiencies, and later on in my career, I attended a hardcore DNA repair and damage conference where I learned about all the 12 different types of DNA damage and repair and synthetic lethalities. And it does take time and work because many of those things we had not learned before as physicians or even scientists. So the question to the 3 panelists are, in the 3 examples that you gave, was there a new skill or a new knowledge that you had to gain to make that successful, and how did you do it?
That is a very good question. I have to say, as a clinician, as a GYN oncologist and a surgeon, and like you said, the timing, it's difficult. I will tell you, I would not have been successful without Martine Felices and, like I said, Laura Benzek and the lab members. And you have to surround yourself like good people. I mean, Jeff Miller, like all the immunology that I picked up along the way, and going to conferences and attending large international conferences, there's no way I could have done this without them. And I think that for me, just being in the position I am, that you have to get a good team. You have to treat your team really well, hopefully, and they will stick with you. And I think we've shown that, and it's really been a successful relationship over the years. And without that, I would never have been able to do this. That's team science for you.
So I had little idea about how to actually translate a finding from the lab, that you vetted the idea in cell lines and in mice. So we approached Dr. Michael Fisher, who's part of the NF Clinical Consortium, a nationwide consortium for clinical trials for NF1 associated problems. And we said, well, what's going to be easier to translate, a new therapy for the benign tumors or the malignant ones? And he's told us the malignant ones. And we said, well, what's going to be the burden of proof before someone would let us try this in people? And he said, you got to show it's active in many PDXs. And we got guided by him really on how to do this. And then the final thing was what makes sense for the design of your first trial. And we decided it would be a window of opportunity, early phase trial, where the patients are only treated for 28 days and they get research biopsies and imaging before and after. And we'd find out by doing pharmacodynamic studies and looking for changes in the tumor if something was really happening. Since this is a rare disease, that all kind of made sense. You're not going to be able to recruit very many patients. You got to learn as much as you can from a small number of patients. All of this stuff I had no idea about really, until I talked to the right experts.
So I think the biggest thing I learned, or the biggest thing that has changed in our world, is just having multi-analyte biomarker availability. So again, when I started breast cancer, we had the estrogen receptor and we could do a charcoal binding assay or stain for it. And that was it. There's one biomarker that you could use. The field has evolved into multi-omic, and particularly in breast cancer, multiple gene expression mRNA, but in other fields, whole genome sequencing or exome sequencing, proteomics is coming, and be able to go from one biomarker to now whatever, thousands of biomarkers per individual patient tumor is a learning field. So how did I manage that? I learned to collaborate with people who really thought about this a lot. One of the things I talked about last time I was here was involvement with the I-SPY trial, but the leader of the bioinformatics biomarker part is Laura Vantveer. Laura was from Amsterdam. She founded Agendia in the beginning, made the 70 gene protein which commercialized the Mammaprint. And she's been very deeply thinking about that. So within that trial, we have weekly calls and we talk about what's happening. And Laura's been great about guiding all of us dumbish clinicians through this minefield of false discovery rates and understanding what's truly real and what isn't. And we have a colleague on the protein side, Chip Petricoin, who came from Lance Leota's lab at the NCI, who does the same thing for the protein side of things. So that's been a big change. While I was thinking about it, I want to comment on one other thing that I learned that I did not understand at all until later, how important the business decisions are around drug development. So as I mentioned, that Genentech drug got turned into a thyroid eye disease drug. But I was invited to go to Genentech to talk about, what was happening in the field, had a nice lunch with their team. And with that lunch was a business guy. And, the business guys told me something I had never even thought about before. And the business guy said, the first thing we can calculate is the minimum economic dose that we have to give for a drug. And so what he was saying was, what's the prevalence of the disease? How much does it cost to make the drug? What kind of overhead do we want to have? And that's a dose that we can understand. And to me, that was shocking. I, as a clinician or as a scientist, you think about, medium effective dosing or toxicity or other things, but business had that sense. And that was something that I actually now took with me into talking about all these guys about, what's the prevalence of disease and how much money can they, that sounds terrible, but what's the margin for anything they produce? And I know as university employees now, we can't say anything bad about the current administration. So I will say one thing, RFK Jr. has a lot of thoughts. And one thought that he's actually right about is, why is biopharma dictating healthcare in this country based on what I just told you? How much money can they make off a particular drug? Why can't we take some drugs that have been repurposed or cheaper, might do the same thing, because there's no financial incentive?
Questions from the audience?
I have a question from Sylvia Balbo online. Is there any aspect unique to the MCC that contributed to make your story possible or successful, or at least contributed to your success significantly?
So maybe each of you could think of the greatest strength that helped you to succeed in your respective projects, if it was something from MCC.
Okay.
Yeah, so this is not a fair question for me, because if these guys can say it's terrible here, and that will fall on me. But so, again, I didn't talk about it this time, but this trial, as well as all the other trials, have to have experts that manage the studies. The trial I talked about last time, the I-SPY2 trial, is very complicated. So building a very robust and responsible and effective clinical trials office now under Dr. Geller's leadership has been absolutely critical to my career development as a translational researcher. I mean, no matter how much you want to do a study, you, the individual physician, and you, your entire clinical staff are not going to do the study because of the special expertise needed to run it. So I say that is, you, for me, was one of the MCC things that has been really responsible for making these studies go forward.
I guess same CTO, close integration with the surgical practice here. So we're able to make our own patient-derived xenografts from MPNST patients. That was super helpful to have some samples that we could test our ideas on, and we still utilize, so.
You know, I think if I look back in the support that the MCCA has given me over the years, like with grant seed funding, the translational working groups in the beginning that Dave Largaespada had started, you know, I think that was my first foray into like being able to see what all the basic scientists did across the university, and bringing that think tank together really was able, you know, I was able to connect with people I would probably never would have been in touch with as a clinician scientist before. So I think that was, you know, really important in the beginning, and then the seed funding to start these grants, and then the grant reviews that people have done for me over the years, you know, where you submit the grant, they review your specific aims page. I think that's been invaluable for me for the Cancer Center for many years.
Yeah, I'll just answer on behalf of myself, even though I wasn't a speaker. Try to take advantage of the strengths that are already here, and don't try to put a square peg into a round hole. When I first came here 3 years ago, I had a lot of ideas where the expertise just simply wasn't here on that particular area. So I remember I approached Justin Drakes, who is one of the proteomic experts in Department of Pharmacology, and I said, Justin, I have this idea for a prostate cancer trial. I want to use a liquid biomarker from the blood, and I want to detect circulating tumor DNA and other nucleic acids. And he said, well, I don't know anything about that, but I know a lot about proteomics. And we have a way of detecting the proteome from extracellular vesicles in the blood from patients. We're sure we can do it. We've never done it before, but we're sure we can do it. And I said, oh, I don't know anything about proteomics. I can't even spell or write mass spectrometry, let alone phosphoproteomics. And I spent the next 6 months learning everything I could about proteomics. And we started that collaboration, even though the starting idea was different. But then we took advantage of Drake's expertise that he had, and he changed the way I thought about it. And I also changed some of the things that he was thinking about too. Other questions from the audience or online?
So this is great, thank you. I'm going to start off by, I'm right now taking some of my consulting time for this second, because I'm wearing my hat as the founder and director of Veterinary Research Associates. I'm not an employee of the university. The current administration is full of stupid people, they're dangerous, and we need to fight back. Okay, now I'm wearing my university hat again. So my question to you guys is it's interesting that a lot of the work that you did was funded by non-government sources, non-NIH sources. And I think one of the things that we only do well when lightning strikes, as opposed to being very intentional about it, is to go after these non-federal funds. So what kinds of things do you think we can do better going forward to work with the powers that be at the university to actually increase the value of that non-federal funding to the university and to the research, so that we can be very strategic about partnerships with pharma, with small companies, and with philanthropy? Because I think there's a lot of money that's left on the table, because when we go after it, the efforts that we put in are devalued, and we don't get the support from the university, and so donors go elsewhere. So how do you guys think we can change the outlook from higher ups?
David, we'll start with you on this one.
Well, thanks for asking that, Jaime. You know, a lot of the money that we've gotten for NF1 related research has been from foundations. So Children's Tumor Foundation, which is the largest, you know, private sort of foundation funding NF related research based in New York. I developed relationship with them early on. I'm always willing to review their grants. Same thing for Alex's Lemonade Stand Foundation and others. I guess the problem is they pay 10% indirect. So I don't know what to tell you, to be honest, because all of you who want to be successful in running a lab will have to have a very diversified portfolio of funding. So you can't just rely on one source. It's going to be philanthropy. It's going to be foundations of various sizes. It's going to be the NIH. So I don't have a great answer for this, this issue. I was kind of wondering if some of them might decide that based on what's happened, in the last several months, they should be paying more in indirects, sort of realizing that they kind of been getting a free ride in a sense. If they're paying really low indirects, it's somebody else that's funding the infrastructure necessary to do the research they fund.
Yeah, I'll just say really quickly, I was on a phone call yesterday with the Minnesota Ovarian Cancer Alliance, and with this exact question was being asked. They have never included indirects, 0 indirects. And now reconsidering that in light of what's going on. And I think, educating these groups and the importance of this is really critical. And as David said, you think about it, even the money that we get from doing these trials, these clinical trials, like my percentage on that is small, right? Like it's minuscule, dollar wise. But but the money that we bring into the institution, and so it's really difficult because we're not recognized for, unless it's a R01, or even the DODs are not looked upon like that way, and that funding has gone to pretty small numbers now too. So I think we have to change the perception of that. And people have tried to do that. And we're running national trials and we get no, credit for that as well either. It's like an R01 or nothing a lot of the time. And I think that's really a hard situation, especially now. Changing that is going to be difficult.
Do you have an answer?
In terms of clinical trials, I kind of have this fantasy world where CMS, Medicare and Medicaid services, would actually pay for clinical trials important to them because it will ultimately determine their costs. Now, some of our colleagues do this, our Future 50 for State Canada. They have more of a universal healthcare system and they fund clinical trials. If you look at the Canadian cooperative group mechanism, a lot of that funding comes out of the money that is allocated to healthcare, because I would argue as a clinician, I think every clinician in this room would argue that participation in the clinical trials is the best healthcare you can get. And therefore, why aren't we paying for that through our healthcare dollars? So that would be one way to alleviate part of the system. Maybe leave the NCI alone and say, well, you're just going to fund fundamental translational work. You're not going to do the clinical trials anymore. We'll put that in another bucket. But that requires significant political will to make that change.
Okay, we've got another question from our online audience. Luana Ward asks, can you talk about your experience conducting your first trial compared to your latest trial? I guess what has changed?
I have to say, I mean, it's when you start this, it's like drinking from a fire hose, right? I mean, there's so many things. And I'll say back then, you don't really know what you don't know kind of thing. And over time, you learn these things and you learn how to become more adept, and the importance of being on all the calls and, reviewing protocols so that the best protocol and how to write it. And I will say, I think the CTO has developed over time too. And we have really good program managers that help to manage, there may be investigators running 10, 15 trials at a time. And it's a significant amount of work, a lot of signing documents and recording adverse events, and that's really critical. And I think that over time the CTO has really learned how to work with our investigators better and closer to prevent any complications. Because obviously the most important thing is the patient and keeping them safe and protecting our investigators. So I think over time that has evolved. And it takes time, and doing the training that CTSI offers and doing these types of trainings for new investigators is really critical.
Yeah, I'd say way back when it was an apprenticeship system. You know, you followed around a senior clinician who did clinical trials and you did what they did. You know, there was no HIPAA training, there was no city training. For those of you that haven't had to do city training, you should consider yourself fortunate. But on the other hand, there wasn't I will say the other piece was, even though that I learned from an apprentice, a guy named Dan Von Huff, who was a phase 1 trialist, even then the trials were less complicated than they are now. So it was manageable by a research staff and maybe one RN for a portfolio of 10 phase 1 trials. It's impossible to do that now because they've gotten so much more complicated.
David?
Yes, thank you all. This is great. I'm coming from the outside, biomedical engineering, trying to learn all these things as I go here. And I was struck by Dave and Doug, both of you mentioning repurposing as part of your story. And it kind of hits to what Doug was also mentioning about the current Secretary of Health and Human Services, kind of identifying this as the cost and what's driving the research agenda forward. Oftentimes revolves around the profit and cost aspects of these things. And there's an opportunity to repurpose drugs that aren't costly. Is that something, if we have enough successes there, could we like be known for that at Minnesota? That could become a thing. We could be a repurposing clinic with the goal of bringing down healthcare costs and having more options available to patients. Or kind of in parallel to that, not to get off topic, but Doug mentioned a drug that was abandoned by a large pharma that he was hoping venture capitalists could salvage somehow. So if there's like a repurposing clinic and a salvaging clinic to kind of like take these things that have been invested in that could pay off and yield relatively low cost, effective solutions, could that become a thing that we are known for and could get funded for by the federal government, even with the current administration?
I'll answer that first and then I'll ask who has other ideas. You know, as a oncology fellow in an institution where I didn't have connections to drug companies as a fellow when I was at Johns Hopkins, my first 3 trials that I wrote were all repurposing trials. They were in prostate cancer. One was repurposing digoxin, one was repurposing itraconazole, and one was repurposing disulfiram antabuse. And one of those 3 trials was successful. The phase 2 study of 40 patients, you know, we saw very interesting responses. The other 2 were not. But the challenge there is you get stuck after phase 2. You have a positive phase 2, you have a high impact publication, and then the study dies because a phase 2 study, just for the audience to know, typically costs between $2 and $4 million, 40 to 60 patients. Then you need to move into a 100 patient, 300 patient, 500 patient study. Now you're talking about 50 million, 100 million. And because those repurposed drugs don't have a pharma company to sponsor the conduct of the trial and to provide the drug itself, because even if the drug is cheap, if you need to buy that for a patient who takes it once a day for 6 months, and there's 100 of those patients, even the cost of the drug. So it's a great way to make your career, but then it doesn't lead to FDA approval. Now it can be used off-label if the guidelines recommend it, but that was my perspective.
No, I don't have a lot to add, but Dave, what you're getting at is how can you make something practice changing? So if Emmanuel demonstrates that a repurposed drug is equivalent to an existing drug or even better, how do you get that to change? And so then you've got to run the large phase 3 trial, and that's out of the. I mean, I think if the state ever said, yeah, we do want to do this, we will fund your phase 3 trial. You know, I mean, you could just think, talking out loud there, the standard of care is the standard of care. If you have an immune checkpoint inhibitor that's a simple drug that exists, I'll tell you a story. There's a reasonably good story for beta adrenergic receptor and anti-cancer chemotherapy. So propranolol, a drug that's been around forever, we tried to put that in the I-SPY, but we just couldn't make the finances work. If you said, well, the state of Minnesota or some other entity will pay for that arm, we could have done it.
Okay, we've got time for one final question from our online audience. Jeff Miller asks, can you each list one pro and one con in working with industry partnerships? And a recommendation in your experience if the pros outweigh the cons or vice versa?
I'll go first, Jeff. You know, my advice is just be true to yourself and be true to the science. Don't be worried about offending these companies. They don't love you. One of my mentors told me, you know, companies are not loyal to you ever, even though they might seem like they are. One of the most eye-opening experiences for me was I was invited to an advisory board. And when I was asked what I thought, I said, this is the worst trial design I've ever heard of in my life. And I thought I'd never be invited back. And then I was asked to be the PI of the trial because they took into consideration what I didn't like about it and fixed it. So be true to yourself. Don't worry about offending them. They're not loyal to you. You don't have to be loyal to them either, actually. If there's another company that's easier to work with, sometimes you can pin one against the other. You can say, well, I'm talking to two companies. Second one, they're making my life very easy. You guys are just being very obstructive. What can you do to match what the other guys have offered me? You know, and sometimes they will up their offer, and then you continue working with them. Play hard to get.
I think, you know, I've worked with the company for a long time, and as has Dr. Miller. And, you know, they, in the beginning, when there was a lot of money and the trials were going great, we were getting the drugs. And then when that switches, then you're in trouble, right? Because you may be halfway through a trial and they don't want to pay anymore. So currently we're using philanthropic funds to fund a clinical trial just because we want to get through this phase. We believe that it may work, but they've moved on to the next best thing. And it's that constant switching that makes it very difficult. And you know, the pro is when the money's there and it's great and they're, you know, you're going on all the trips that Doug said. And then the minute that changes, you know, you're in halfway through a clinical trial and they'll pay for the drug, but they're not going to pay for anything else. And so that is one of the cons. It can be difficult navigating that.
So I guess the pro is that because we work on rare diseases, there are pharmaceutical companies out there that are dedicated now to orphan diseases and other things. So SpringWorks, Alexion, others that we've worked with, Day One Pharmaceuticals. So that's great, and they're really looking for these people with this rare expertise. And so SpringWorks has been great to work with. I think the con is that the whole process is very opaque to someone like me. So the trial that we opened was originally, we spent a lot of time trying to do it with Alexion and selumetinib plus fernistat, a different MEK inhibitor. And the whole thing, Alexion scientists wanted to do it. And then it got reviewed at Merck because they had some rights that I still don't understand, because there's a relationship between Alexion and Merck killed the idea. So we had no idea why. It was almost like it's the mafia, you know, like it's not happening, you know, you don't really need to know why, or we can't tell you why. So luckily SpringWorks did work with us. So anyway, it's just that it's opaque. And so I know it had something to do with what Doug was saying, the business issues that were of concern to Merck. So I just don't get it.
Yeah, I know we're over time, so I'll just echo what Emmanuel said. The pro is companies are listening to us, the academics, more in their trial designs. The con is I think a lot of business decisions are made about the future of a drug that even the medical team from the company you're working with have no input into. And I think that maybe is what you ran into, David.
All right, I'd like to thank the speakers in the audience. I hope you had fun.